Hi everyone, my name is Jeroen, co-founder and CEO at Semarion, and I would like to share with you what we are doing to revolutionize cell-based assays. We have developed a truly unique microcarrier platform which brings adherent cells into suspension. This is particularly useful for drug screening programs where features such as assay automation, assay miniaturization, and assay multiplexing would enable faster and better data generation. Our materials are made using microfabrication technologies and leverage the latest advances in nanomagnetism and smart materials. These carriers are furthermore designed to seamlessly integrate with existing workflows and do not require bulky new equipment. And while we are bringing consumables products to the market, we are also engaging in technology partnerships to develop bespoke solutions for challenging cell screening setups. So, we focus specifically on adherent cells. Cells which need to be attached to a surface or a substrate. Assays on these cell types dominate drug discovery, where they are used to evaluate compounds for potency, toxicity, and mechanism of action. And while workflows with suspension cells have enjoyed liquid handling and miniaturization improvements through microfluidics and other flow-based systems, adherent cell assay workflows have not seen similar gains. The key problem is that adherent cells need to be attached to a culture or well plate. And this gives rise to three broad challenges. The first one is that assay preparation times are long. Before you can run an assay, the cells need to be cultured, collected, and distributed into multi-well plates. You then need to wait for the cells to reach the right confluency and morphology, and this whole process can take multiple days and severely limits the rate of data output. It also leads to further issues with data reproducibility, as these cells are typically prepared on multiple days by different people. The second challenge is that cell multiplexing, or combining various cell types into one well, is currently not possible. This drastically limits the efficiency for drug screening programs where large panels of cells are used, making it incredibly time and cost intensive. And the third challenge is that there's always been sort of a drive to miniaturize assays, but the miniaturization limit has been reached with the 384 well plates for cell assays. This means that assays with scarce cells, such as stem cells or patient-derived cells, can currently not be run at scale. Our solution is simple. Keep colonies of cells attached to a surface, but now bring the surface into suspension. We have been able to do exactly that through our Semicide microcarrier platform. By turning adherent cells into liquid reagents, we can drive 10x efficiency gains in drug screening and produce better data faster. These semicides are essentially ultra-miniaturized, steerable wells. We can sort them, tilt them, and redeposit them at the bottom of normal well plates. The semicides provide unique advantages. We can now move and freeze cells while retaining their adherent morphology. This reduces assay preparation times by more than tenfold, while also increasing longitudinal assay reproducibility. We can incorporate barcodes to enable true cell multiplexing. When we multiplex 10 cell types, for example, we can increase the efficiency of the screening campaign by tenfold. We can further miniaturize cell assays by choosing how many carriers and cells we add per well, and this means that we can add 10 times less cells to enable broader screening campaigns with scarce cell tissues. One of the main benefits of our approach is that we can seamlessly integrate with existing workflows. This allows users to keep working with their existing equipment and infrastructure while benefiting from the features of the Semicide technology. Our microcarriers are immobilized on discs, like the one you can see here. Cell seeding can be done as usual by simply pipetting cells on these substrates. Once the correct confluency and morphology is achieved, the semicides can be lifted off the disc and brought into suspension for direct use or long-term storage. To run an assay, cell-containing semicides are titered into a well plate, and our magnetic plate holder pulls the microcarriers to the bottom of the well while the cells are facing upwards. At this stage, users can run any type of assay they are used to, including plate reader-based or microscopy-based assays. Our semicide carriers are made using microfabrication technologies, typically used to produce computer chips. This allows us to control the size and shape while incorporating various active layers with sub-nanometer precision to tailor their features. We start with a smart release polymer, which slowly dissolves in cell culture media to release the carriers after cells have attached. The structural photopolymer is used as the backbone of the carriers and controls the shape and incorporates optical barcodes, such as QR codes, for example. And our magnetic heterostructure facilitates truly unique magnetic properties. We can not only sort and immobilize these carriers, but also orientate them in 3D, all while preventing them from aggregating and clumping together. The materials are rendered biocompatible through a gold cap, to which a final cell attachment polymer is added. Here you can see cells on carriers before they are released. This is a bright field image of carriers which have been redeposited into a well. And this clip shows how we can move, rotate, and redeposit the carriers using an external magnetic field. These are live cells which have been labeled with the viability dye AM calcine. We will enter the market with our Semachip. This basic product allows users to load cells of interest, freeze them, and deploy them for plate reader-based assays. Freezing cells in their adherent morphology can be truly groundbreaking, specifically for compound profiling and structure-activity relationship studies. During lead optimization, for example, chemists work to create novel compound iteration. Once they are done, they typically ask cell biologists to evaluate these iterations. Currently, that means that they will need to culture enough cells, strip them, plate them, and wait for them to reach the correct morphology. This process can take a long time and delays the drug discovery process. With the Semachip, users can load and freeze batches of adherent assay-ready cells. They can be used on demand, completely removing the lengthy cell preparation times. This does not only drastically accelerate the decision-making process, but also reduces batch-to-batch variability often seen with adherent cells. We have tested this product with external organizations, namely the Milner Therapeutics Institute and the Physics of Medicine labs at the University of Cambridge. We asked our trialists to run a dose-response drug incubation experiment with human kidney cancer cells on semacytes. Compared to the normal well plate-based experiments, these cells on semacytes showed similar morphology. Their viability post-stowing was also comparable. The final output of these experiments, run at different centers on different equipment and at different times, yielded identical IC50 values and assay outputs for both the semacyte group and the well control group. In this setup, we were able to showcase a 50% reduction in experimental time while using 5 times less cells per well. Another solution which we are currently developing is our Tentplex product for imaging endpoints. Here we will use sets of optically barcoded semacytes which can be loaded with different types of cells. By combining 10 cell types into one well, we can drastically reduce the cost and increase the speed of screening campaigns which require large cell panels, for example, drug positioning or IND enabling studies. Our basic software can identify semacytes from images and deconvolute the barcode for straightforward image analysis. We have validated a variety of phenotypic and viability-based endpoints, both on live and fixated cells. Finally, we're also offering bespoke application development for specialized cells, assays, and equipment. Through technology partnerships, we develop niche solutions to enable novel research methods for our biopharma clients. This is particularly relevant for patient-derived cell assays. Currently, one biopsy would yield enough cells for roughly half a 384 well plate. But with our semacyte ultra-miniaturization approach, we can increase the breadth of an assay by over tenfold to obtain more insight from these biologically relevant cell types. A final word about the company and the team. Semarion is an early-stage spin-out from the Cavendish Laboratories at Cambridge University. As a company, we bring together various scientific disciplines to solve complex biological problems. I myself have worked for the last 10 years on cell model development and screening of various drug modalities such as RNA therapeutics and stem cells. Jeroen is our CTO and has focused his academic career on the development of nanomagnetic materials for biological applications such as tumor ablation. Our chair Paul is CBO at Mission Therapeutics and has vast management, deal-making and scale-up experience. Ornette Russell is a professor of experimental physics and a fellow at the Royal Society. And Ornette Dell was co-founder of Essendon Biosciences and developed a successful IncuCyte cell screening technology. If there is any interest in testing our materials, discussing partnerships, or joining our team, feel free to reach out. And together, we will usher in a new era for cell-based assays. Thank you.